ready
Okay, thank you.
to be in. Thank you.
Thank you. So now we are online and we are live on YouTube.
Namaskar, Dr. Chogle. I would be over here joining online.
हाँ शुरू कीजिए.
I can hear you, sir.
Yeah.
I'm very much there and much honored by your speech. Thank you so much.
I think you have to introduce the first speaker. मैडम आप फर्स्ट स्पीकर एंड टॉपिक इंट्रोड्यूस कर देंगे. Hello, madam, is it audible? I think it's not audible. Not audible. It's not able to. Madam, it is audible now to you?
Yes.
Okay, now you can just introduce the first speaker so that we can start our session.
I'll request Dr. Chogle to please invite the speaker because I do not have the material, the CV of the speaker. Kindly.
Thank you, Professor Madhubala. The delegates, the first session of this international conference on implications for management of cancer is beginning. Along with me, Professor Madhubala, the president of the Indian Society of Radiation Biology, is chairing this session. And the first talk is from Dr. Seema Gupta, who is a scientist at Washington, D.C., USA. And she will be speaking on the promise and challenge of combining radiation therapy with immunotherapy, because it is very, very important that radiotherapy needs the combination of other therapies. And she, from her experience, will be talking on the challenges in radiotherapy with immunotherapy. So now I hand over the floor to Dr. Seema Gupta from the scientist Washington DC, USA, to start her talk, and then she will get 20 minutes for her talk. It is Dr. Seema Gupta.
Hello everyone. Today I'm going to talk about the promise and challenges of combining radiation therapy with immunotherapy. Before I do this, I would like to thank the organizers for giving me this opportunity to talk about this important topic of the combination of radiation therapy with immunotherapy. So if we just go back by 15 years, we see that the chemotherapy and radiation therapy were the major clinical remained the standard for anti-tumor therapies, but there were a few clinical trials which were going on with vaccination and other checkpoint inhibitors in 2007. But if we just go by a decade, if we just advance by a decade, we see that in addition to chemotherapy and radiotherapy, the immunotherapy, especially the checkpoint blockade with anti-PD-1 and anti-CTLA, have also become important pillars for cancer therapy. And nowadays, several clinical trials are trying to combine these strategies together to further enhance the response, anti-tumor responses. And even the preclinical experiments combining these two strategies have been, these two or even with chemotherapy, have been, the number of these trials have increased significantly. And we can see that there are thousands of trials ongoing just with one of the checkpoint inhibitors, the pathway which target PD-1, PD-L1 target in both the solid tumors and in the advanced, and also in the hematological malignancies. However, we still remain away from the maximum, the optimum response which we expect when we combine these, when we give immunotherapy. And here we can see that there is a prolonged survival when we give immunotherapy compared to the standard therapy, but the number of patients who respond to immunotherapy is still very low. And there is a gap here, like big gap here, which this curve should go from here to here so that we are closer to the cure. And several mechanisms have been suggested for resistance to anti-PD-1 therapy or immunotherapy. And one of the important innate mechanism which is being described is somatic mutational load. Like here in this example, where the BRCA where the tumors have BRCA2 mutations, they respond much better to anti-PD-1 therapy than the patients who have tumors with no BRCA2 mutations. And in addition to the innate resistance mechanisms, several acquired resistance mechanisms have been described, such as antigen presentation, tumor-mediated suppression, and inhibitory checkpoints, etc. To overcome these resistance mechanisms, scientists have tried to combine these the immunotherapies together. Like here, the PD-1 blockade combined with anti-CTLA could significantly improve the survival of the patients from 20 to 30% to 50 to 60%. However, we are still not near to the cure, and it needs further combination strategies with these immunotherapy to reach about 100% survival. And here in this presentation today, we are going to see if X can be radiation therapy, and if it can be, then what are the problems or challenges, and what are the issues we still need to investigate before we go ahead and combine these together with so we know that radiation therapy has both immunosuppressive and immune stimulating effects, and due to the secretion of the several immunosuppressive cytokines through the TAMs or through the Tregs are recruited, and these Tregs also secrete immunosuppressive cytokines, leading to the suppression of the cytotoxic T lymphocytes and dampening of the immune response. And after radiation therapy, at the same time, there can be the release of several DAMs and the inflammatory cytokines such as TNF alpha, interferon gamma beta, etc., which leads to reduced tumor proliferation and increased tumor regression, and DC recruitment and maturation of the DCs, leading to activation of the CTLs through several chemokines. And these CTLs release interferon gamma, TNF alpha, and several enzymes, granzymes, and perforin, or have cell-to-cell interaction, leading to apoptosis of the cancer cells and therefore increased anti-tumor effects. In addition to the immunosuppressive and immune activating effects of the radiation therapy, there are abscopal effects of the radiation therapy are also very well described, and which happens because when the irradiation was when the tumor cells were irradiated, there is either release of the new antigens, there is release of the DAMs, and there is release of the inflammatory cytokines, all leading to the maturation of the dendritic cells and antigen presentation to the T cells in the lymph nodes, leading to activation of the CD8 T cells, their proliferation, which can affect, lead to the local tumor control, as well as it can also kill the tumor cells in the non-irradiated metastatic tumor, leading to the abscopal effects of radiation therapy. So these two important characteristics of the radiation, the effects of the radiation therapy, are important when we combine radiation therapy with immunotherapy. Of course, there are additional radiation therapy induced anti-tumor effects, but these two are the major effects of radiation therapy which we are going to discuss today. And so of course there is a promise of combining these two, and then there are several hundreds of preclinical studies that have been conducted in the last several years. But here I'm just going to show the few of the clinical trials where this promise has been shown, or where when combination of radiation and immunotherapy has shown promising results. So this is a case of hepatocellular carcinoma, where the patient shows significant immune response when immune-mediated anti-tumor response when SBRT is combined with immunotherapy. There is significant reduction in the tumor size and complete resolution of the lung metastasis. And these are the example from two other patients where the tumor size is significantly reduced when SBRT was combined with. In another clinical trial of where the mucosal melanoma patients were treated with ICI alone or radiation therapy alone or their combination, there was a significant control increase in the control rate of the target lesion when the combination of ICI and RT was given compared to the monotherapies. And here also we can see that there is a decreasing tendency in the target lesion volume in patients with mucosal melanoma when ICI was combined with radiation therapy. So there are some trends and some response. However, when we look at the progression-free survival or overall survival, these differences remain insignificant between the monotherapies and the combination. And not only these results, but the other trials also show that there are not significant differences, although there is some improvement in the response. This shows like another example from Pembro-RT phase 2 randomized clinical trial in non-small lung cancer patients, where PFS and overall survival remained insignificant between SBRT, SRT combined with the immunotherapy or immunotherapy alone. And this is another example in head and neck squamous cell carcinoma, where SBRT given in 3 fractions of 9 gray remained insignificant when overall survival was compared. So this shows that there are challenges when we combine radiation therapy with immunotherapy, and we are not able to get the optimal results. And recently, based on the several studies conducted by several groups, we reviewed the literature, and also based on our studies, we came up with some challenges and critical questions that should be considered when we are trying to combine radiation therapy with immunotherapy. And also these are the points which need to be taken care of and also investigated in the near future, so that we have optimum response of the combination therapies. And of course, the most important is the radiation, the quality of radiation, whether it is high versus low LET radiation. And now we have flash, we have microbeam, we have so many other types of radiation. Then the dose of the radiation, the fractionation, whether it is going to be low dose or high dose fractionation, the dose rate and the schedule, and also whether we should irradiate the whole tumor or only the partial volume tumor irradiation is sufficient, so that we have reduced normal tissue toxicity also, or maybe we have spatial radiation therapy mediated effects when we have only the few parts of the tumor are irradiated. And indeed, the immune characteristics of the tumor cells are also very important, especially how much checkpoint expression the tumor cells have and their immunogenicity. And also based on the susceptibility of the tumor cells to the immunotherapy, the type of immunotherapies which need to be combined with the radiation therapy needs to be considered also. And as in the beginning, I showed that both the radiation therapy can induce both the immunostimulatory and immune suppressive effects based on the what kinds of cytokines are released, whether these are immune activating or immunosuppressive, and what factors dictate the balance of these. If we understand those factors, then we are in a better situation to combine radiation with immunotherapy to have optimum benefit, or to have the maximum benefit. And some other points need to be considered, like lymphocyte sparing, because we know that lymphocytes are very sensitive to radiation. So this needs to be taken care of. And I'm going to show like how lymphopenia may affect the RT-mediated response in the combination therapy. And also the right metastatic site needs to be identified. And as I mentioned, like spatial fractionation may be affecting the immune priming, like if we use lattice therapy versus we are using the whole tumor irradiation. So to quickly show you some of the how some of these challenges or these factors influence the response after combination therapy of radiation with immuno, I'm going to quickly show some examples here. So here we can see that immunotherapy, when combined with SRT, is able to enhance, like this combination has much better survival than either of them alone, and external beam radiation therapy was not able to enhance have synergy with the immunotherapy. So the type of radiation given is affecting the response here, and SRT, independent of the systemic treatment we are given here, is able to enhance the response, whether it is chemotherapy or immunotherapy. And again, the low bed versus high bed also affected the response. And when the radiation was given as high bed, more than 60 gray, then the responses were much better either with immunotherapy or with chemotherapy compared to the low bed radiation therapy in combination with the chemo or immunotherapy. And as I mentioned about the lymphopenia, here Grasberger et al. Showed that in various types of the tumor, the odds ratio was more. Hearing is one of the important parameter or endpoint to be considered when we are combining radiation with immunotherapy. And this figure shows the type of radiation, like photon versus proton. And we all know that when we give photon based radiation, there is so much low dose of radiation which is given to the surrounding tissues which are surrounding to the tumor, and which may include even the arteries which supply the blood, leading to lymphopenia, while the proton based irradiation may not have these kind of effects. And also the dose rate can influence how much lymphopenia we are generating, because at the high dose rate we may be able to give higher doses of radiation but with less exposure of lymphocytes and to the radiation, and therefore sparing majority of them. I would also like to talk about another important question which we need to consider when we are combining immunotherapy and radiation therapy, and that is the scheduling of the two therapies. So we recently published a paper in Nature Immunology in 2019, where we showed that the sequencing of PD-1 with respect to the antigen priming is very important for anti-tumor response. So since we showed that when we are giving vaccine and PD-1 together, we have very good anti-tumor response in a cold tumor model of TC1. But when we give anti-PD-1 before antigen priming, like one additional dose of anti-PD-1 before the antigen priming, we lose this response completely, and which can be seen here also in survival. So here we have very good response at different doses of anti-PD-1, but when we give anti-PD-1 before antigen priming, the response is lost, the green lines here. And the first thing we looked into is the tumor microenvironment for immune repertoire, and we found that both the CD8 T cells and the antigen-specific CD8 T cells were significantly reduced when the anti-PD-1 was given before antigen priming. So just focus on the last two bars because of time scarcity. I'm going to quickly go over these results. And we found that even after the first vaccination, if you look at the total CD8 T cells or antigen-specific cells, that these cells were, the antigen-specific CD8 T cells were reduced, which shows that these responses were generated early during the treatment schedule, so when anti-PD-1 was given before vaccination. So we compared these two time points, and we found that there was increased apoptosis of the CD8 T cells. There was increased apoptosis of the antigen-specific CD8 T cells when PD-1 was given for vaccination, and these cells were less active, as shown by the CD40L expression reduction here. And these cells were also less functional because interferon gamma production by these cells was much less when anti-PD-1 was given before antigen priming. So these data show that sequencing of antigen priming with vaccine and anti-PD-1 is crucial for the success of the combination therapy. And vaccine, when given together with anti-PD-1, it reverses the anti-PD-1 tumor resistance, but prior PD-1 blockade abrogates the vaccine-induced susceptibility to anti-PD-1 therapy. And we found that when we give anti-PD-1 before antigen priming, there is increase in the PD-1 CD38 high cells in the tumor microenvironment, and these cells have been shown to be dysfunctional cells, which are not reprogrammable cells. They can't become functional again once they are generated. And we found even antigen-specific cells were significantly, PD-1 CD38 high cells, dysfunctional cells were significantly increased when anti-PD-1 was given priming. Their activation was less, as seen by CD40L, and they had increased apoptosis also, so they were dying much more. And we wanted to confirm that these cells are indeed responsible for reduced or resistance to anti-PD-1 therapy when it is given before antigen priming. So indeed, when we depleted these cells from the total CD8 T cell culture, we had better response when the total CD8 T cells were infused into the tumor-bearing mice, which shows that indeed these cells are the ones which are responsible for resistance to anti-PD-1 therapy. And we also found that when anti-PD-1 is given in the cold tumor-bearing animals like TC1 tumor without any antigen priming, number of these cells was significantly increased with increased apoptosis of the total CD8 and the antigen-specific PD-1 CD38 high cells. And indeed, we found in the patients also that in the post-anti-PD-1 therapy, the number of these cells were significantly higher in the non-responders than the responders in the microenvironment, and also in the blood, these cells remained high or at the same level during the therapy in the non-responders compared to the responders. And interestingly, even in the base samples of the tumor, like pre-therapy samples, the non-responders had higher number of these cells than the responders, which shows that these cells can be the predictor for failure of anti-PD-1 therapy. So these results basically what I showed shows that suggest that the anti-PD-1 should be given after proper priming of the T cells with the antigen, especially at least for the cold tumor models, at least for the cold tumors in the patients. And we know that radiation therapy is one of the mechanism or one of the strategy in addition to chemo and other strategy that can heat up the tumor, means make the tumor inflammatory or make the tumor infiltrated with lots of immune cells. So it seems that we should give radiation therapy before immune checkpoint inhibitors, at least for cold tumor models. And indeed, one of the studies by Cabilzo et al. Showed that it is based on the mice studies, shows that the optimal timing of checkpoint blockade appears to be in the early phases of the radiation therapy, so that we get maximum benefit of the combination therapy. So based on all the points I explained above, recently we came up with a few of the recommendations and suggestions based on the type of the tumor, whether it is cold tumor, hot tumor, or the metastatic site, that what kind of dose or whether it is partial radiation therapy or spatial fractionation radiation therapy, or it is a hybrid, and what kind of fractionation, and whether it is what kind of schedules should be given based on the type of the tumor. Of course, there are several factors which I already explained in deciding the dose and fraction, such as the lymphocyte sparing, selecting the right metastatic sites and number of the sites for the RT, intratumoral boost, impact of underdosing, normal tissue protection, and other aspects of chemotherapy given with radiation therapy, because in most of the cases still we give chemotherapy to the patients. So that also need to be considered. And so overall, I would say that there are multiple factors that influence the RT's immunogenicity, and dose, fractionation, and schedule should be tailored to the characteristics of the tumor and the immunotherapy used. And all these factors which need to be considered for the optimal response for the maximum benefit with the combination of radiation therapy and immunotherapy, I already explained in the previous slide. So these need to be further investigated and both in the preclinical and clinical trials before we combine them with confidence to get the optimum benefit. With that, I would like to acknowledge the Khalif lab where I work, and several of our collaborators from Massachusetts General Hospital, from Memorial Sloan Kettering Cancer Center, from University of Texas, and also the biostatistician who has done several of the statistics in this which I showed in the Nature Immunology paper, and also the antibodies were provided by Scott Hammond. I would like to thank all of you with this, and if you have any questions and comments, please write to me at sg1335@georgetown.edu, and I wish you a very good remaining day and a very good conference. Thank you again. Bye-bye.
lost contact.
Complete response. So radiotherapy has its limitations also.
No, sir, video has completed. This lecture is completed. Kindly invite other one.
I'm giving just the comments about the options. So immunotherapy is a very
तो ये क्या बोल रहा है? Video is disturbing.
given a very nice overview of their study, that how this immunotherapy. She talked about different modalities of proton therapy and particle therapy and other things, because radiovalent responses are different. So ultimately, basis of radiotherapy is radiobiology understanding. That what she has shown. A lot of research in this area is needed. So, Madam, do you want to add anything from your side?
No, you have very rightly, sir, very rightly summarized this talk by Dr. Seema Gupta. And this is really a point to ponder whether we should recommend this or we still carry out more radiobiological studies, before taking it to the clinics. Because immune response is something which is very, very personal to an individual. And radiotherapy, which we have been giving, is a generalized treatment. So here comes the dichotomy in how to take it forward in the clinics. Therefore, what my suggestion is, sir, if we can actually take this forward, we should be allowed to take the biopsies of the individuals. If we have information on the baseline immune response and then its modification, probably it is going to be if we analyze those samples and generate data, it's going to be a huge contribution to the society. I personally feel we should share this data. We make a repository and we share this across the world, because otherwise this is a never-ending problem. Thank you so much, sir.
Truly, this is a multidisciplinary approach, and interdisciplinary research inputs are very, very important. It is a tailor-made, evidence-based treatment, and nowadays laboratory they are finding that the tumor tissue is taken out, and with the flow cytometry and other things, how with the treatment it can respond. So you can predispose before the treatment what you are going to get the response. And that is what we researchers, clinicians should come together for the humanity, for conquering this disease by combined efforts. And in this direction, this research is a wonderful research. It should be carried on. So now we can go for the next talk. So this is from Professor Dr. Rao Papelini, who is a very, very active and very huge contributor of the Indian Society of Radiation Biology. And in each of every activities he participates where it happens in India. But because of this COVID, he is not able to make it. But as I told you, that he is not present in person physically, but by his heart and by his mind, he is with us, and he has sent his message that I am delighted that Department of Zoology, Government Dungar College, Bikaner is organizing an international conference on radiation biology. The international research conference with the objective in bringing together leading academic scientists, researchers, and clinical fellows to the exchange and share experiences and research results on all aspects of radiation biology is pivotal, especially with the rapid development in the field of current times. This interdisciplinary platform for researchers, policymakers, and educators to prevent and present and discuss the recent innovation, trends, and concerns are critical for the rapid implementation of error-proof and cutting-edge technologies in cancer radiation therapy. I appreciate Dr. Purohit and his team for all the efforts to organize such events that not only develop current science and medicine, but also build a new breed of young scientists ahead. I wish grand success for the conference, Dr. Rao V.L. Papelini, and he has his huge laboratory under which a lot of people are working. He has a lot of developments. He has done it. And again, the message to the youngsters for the innovative research in the field so that we conquer the emperor of all diseases, cancer. Because cancer is an emperor of all diseases. वो लाठी से या stick से नहीं मरेगा. We need all kind of weapons and gadgets, all kind of combined therapies to conquer this emperor of the diseases. So I appeal everybody, and that is what interdisciplinary research is. ये नहीं होना कि मेरे अंगने में तेरा काम, सभी के अंगने में सभी का एक दूसरे में काम है. Because cooperatively we can do and we can help each other for the doing good for the humanity. So that is a nice effort his lab is doing, and the speaker, all the three speakers which they will be delivering, they are from his lab. So this is Dr. Krishna Koka. He's a fellow program in Papenevi Labs, what I talked about the Papenevi in Brantford City, USA. And she will be talking on overview of external beam radiotherapy. When I talk about the 125 years development, I'll say what external and then brachytherapy and other things. So now it is over to Krishna Koka for her talk on overview of external beam radiation therapy.
Hello, everyone. My name is Krishna Koka, and I'm a first-year student at Pennsylvania State University in the Eberly College of Science. I'm in the school's accelerated 7-year BSMD program with Sidney Kimmel Medical College. Research on this project began in July 2020, the summer before my senior year at Hemet High School. Currently, we have submitted our write-up to Dove Press for publication and are eagerly awaiting more news. Emerging Technologies in External Beam Radiation Therapy, shortened to EBRT, an indispensable tool for cancer treatment. EBRT is a broad area cancer treatment. It blocks cancer cell proliferation and results in their death through damage to their DNA, cell cycle arrest, cytogenetic damage, apoptosis, and senescence. It minimizes damage to normal cells as they heal quicker than cancer cells. Photon and electron beams are the main form of radiation used in this therapy because they have a low power of penetration and are useful in treating tumors close to the surface of the body. Particle radiation is also used in EBRT, as it is very effective in treating deep-seated tumors by allowing for better dose distribution. There are many types of EBRT, including three-dimensional conformal radiotherapy, intensity modulated radiotherapy, volumetric modulated arc radiotherapy, and more. Each individual modality has a specific advancement and adaptation for the tumor type and spatiotemporal dynamics. We will talk about a few of these throughout this presentation today. Three-dimensional conformal radiotherapy, abbreviated to 3D CRT. 3D CRT is a form of EBRT that uses 3D treatment planning, multiple cross-firing with carefully shaped fixed fields. 3D CRT is an improvement on 2D imaging, as it uses CT imaging instead of X-ray imaging, allowing accurate tumor localization and shows the critical normal organ structures in the imaging, facilitating the optimal beam placement and shielding to be used. It is used to treat a wide range of cancers, including brain tumors, breast cancer, gastrointestinal cancer, lung cancer, and gynecologic malignancies. 3D CRT has been found to enhance the quality of life and promote the prognosis of patients with nasopharyngeal carcinoma by improving short-term response rate, reducing mouth dryness, and reducing parotid gland injury. Still, there are some drawbacks. Patients with endometrial cancer showed higher incidence of GI toxicities post 3D CRT treatment compared to IMRT treated patients, which we will discuss about next. There are also some difficulties associated with 3D CRT related to performing correct quality procedures, positioning, imaging, contouring, dosimetry, follow-up, and dose delivery. Intensity modulated radiotherapy, IMRT for short. IMRT has a unique feature of regulating the intensity of each beam in order to determine the target and surrounding organ shape through computer-based optimization algorithm assistance. This form of EBRT makes use of multiple beams for radiation in order to allow for multiple areas of varying radiation intensity. IMRT has been successfully used to treat head and neck, prostate, breast, lung, brain, gynecologic, and GI cancers, with minimal toxicities to the adjacent tissues and organs compared to the target volume. In studies of patients where the surgical removal of tumors is not possible, IMRT has frequently been used for treatment for non-small cell lung carcinoma, improving the targeting of tumor volume without increasing doses to organs at risk, and even reduces dysphagia. IMRT is prone to geometrical errors compared to 3D CRT due to a higher dose conformity indices, including the considerations in patient immobilization for determining the treatment margin and day-to-day changes in the target volume of some cancers such as prostate and cervix. Volumetric modulated arc therapy, VMAT for short. VMAT is a form of EBRT that provides a full 360-degree of beam directions with entire dose volume delivered in a single rotation by regulating dose rate and multi-leaf collimator, which requires regulation because they affect treatment time through increasing the number of beams, directions, and increasing the monitor units. The rotational method used by this therapy results in differing dose distributions to different parts of the tumor. VMAT decreases the organ risk when compared to IMRT due to this differing dose distribution, which results in lower dosages to the surrounding tissue. As a result of lower organ risk and better homogeneity, VMAT treatment plays a significant role in an uncomfortable immobilization. VMAT uses a simultaneous integrated boost technique. This technique played a large role in the technique having been shown to achieve high local tumor control and increased survival and well-tolerated toxicities in NSCLC patients who are not fit for standard definitive therapy. The potential risk associated with VMAT is the increase in the low dose radiation to the surrounding tissues and organs with a greater chance of having secondary malignancies. Therefore, patient follow-up for a longer period is recommended to estimate the chances of having the secondary malignancies. Stereotactic body radiation therapy, in short, SBRT. SBRT is a new EBRT technique that is made possible by improved radiation accuracy and improved therapeutic ratios for many tumor sites. This technique delivers precise and very high individual doses of radiation by focusing multiple radiation beams to the target from different directions. This delivery occurs over a few treatment fractions to remove primary and oligometastatic tumors that are small, well-defined, and found at different locations in the body. The high radiation dose results in a likelihood of damage to any tissue immediately adjacent to the target. Although this likelihood exists, the small and non-eloquent amount of normal tissue makes this clinically significant toxicity low. SBRT can be used in many types of tumors, including prostate, head, and neck. This technique attempts to take advantage of the potentially low alpha beta of prostate cancer. This technique attempts this by delivering greater doses per fraction to the prostate. In whole gland boost therapy, SBRT is used as a method of delivering a boost of radiation to the prostate. This is accomplished after patients with intermediate and high-risk disease undergo more regional radiotherapy investigation. This technique has shown to be effective in the treatment of early non-small cell lung cancer for patients that were unfit for surgery. Particle radiation therapy. Particle radiation, like proton beams, one of the newer forms of particle beam radiation, has allowed for better dose distribution in deep-seated tumors due to Bragg's peak. Bragg's peak makes proton beams special in clinical use when applied to pediatric and adult tumors near critical structures, including the spinal cord and skull-based tumors. Proton beams are critical here because maximal normal tissue sparing is observed. Particle radiation has a higher biological effectiveness and are very effective in radioresistant cancers, including sarcomas. While particle radiation may be more effective, it has an innate limitation. The production of particle radiation therapy is much more expensive than the production of photons and have more logistical requirements, thereby preventing its wide use in low-income countries. New avenues in EBRT. Recently, the National Cancer Institute has initiated the program for encouraging development of technology and combinatorial treatment using external radiation, X-rays or particle beams used for radiotherapy, for local drug activation or release systematically or intratumorally, delivered therapeutics, including high atomic number elements that emit Auger electrons. This combinatorial approach would deliver safe doses of external radiation dose to locally activate or release the therapeutic agent and prevent adverse toxicities in the normal tissue. By doing this, the range of PDT to deep-seated tumors that are currently intractable with existing PDT would be extended. The figure shown is from Papinini Lab and Krishnan Lab and features nanoparticle-based photodynamic therapy. New avenues in EBRT continued. Furthermore, there's a promise of a multivalent drug to augment multi-pronged cellular damage, including site-specific DNA radiation damage, and also counter infections persisting in several cancer cases. Further down the line, there is hope that the involvement of advanced robotics and artificial intelligence will enhance the utilization of EBRT for effective management of refractory deep-seated tumors. Summary. EBRT is a crucial part of our arsenal of cancer treatments. It has technologically improved significantly related to the in-depth planning and treatment with main focus of each method to image and delineate accurate tumor volume, irradiate maximum target volume, sparing the surrounding normal tissue. Image-guided radiotherapy, stereotactic radiotherapy, and particle therapy would involve the combination of CT, PET have revolutionized with the treatment strategies, leading to enhanced patient survival and therapeutic gain. Involvement of advanced robotics and artificial intelligence will enhance the utilization of EBRT for effective management of refractory deep-seated tumors. I'd like to end off by thanking you all for listening to my talk today, and also by thanking Dr. Papinini, Dr. Verma, and Dr. Durkanath for being amazing teachers during my entire internship. Thank you all, and have a great rest of your day.
Thank you, Dr. Krishna Koka, for giving an overview of external beam radiotherapy, starting from just square or rectangle field to conformal to IMRT to SPRT and all kinds of things, and also the particle therapy. And these all technologies and treatment are available in India, except the proton therapy, the IMRT, IGRT, CRT is available in Bikaner Medical College Cancer Hospital, that is there. Also, when external beam therapy is given, there is a chance that the normal tissue, despite of all precaution, gets radiation, and therefore they have developed some kind of this photodynamic therapy that the drug can go there, accumulate into the tumor, and then when you do external therapy, maximize the effect of that thing. So thank you very much, Krishna Koka, for your overview, and I request Dr. Madhubala to add a few comments or words for this talk.
Thank you, sir. But I have nothing more to add. It was a nice overview. And rather, I must say it's very inspiring for the young scholars, who are coming up, and they are taking so much of interest, and they're going into depth in these subjects. So maybe the young faculty sitting over there should draw an inspiration. Thank you so much. We can proceed to the next.
So now we move on to the next talk of this Papinini group is Dr. Sahiti Madireddy, and he is SIR Fellow Program in Papinini Lab in USA, and we'll be talking about overview of brachytherapy. You'll listen to the external beam radiotherapy, brachytherapy where radioactive sources are kept in close contact with the tumor or inside the tumor, that he'll be talking. So now I hand over the floor to Sahiti Madireddy for his talk on overview of brachytherapy.
Hello, everyone. My name is Sahiti Madhuredi, and I'm from the Massachusetts Institute of Technology. My gratitude to the organizers of the International Conference for Radiation Biology. Today, I will be presenting work done at the SAR Fellows Program with Papanini Labs, and my talk is Technological Advancements in Brachytherapy for Cancer. The previous presenter went over external beam radiation therapy, or EBRT. Brachytherapy, on the other hand, is a short-range internal beam radiation source located close to or inside the tumor. It can be either a temporary or permanent treatment, and it can be used as a monotherapy or in conjunction with surgery and EBRT. The main advantage of brachytherapy is that it can safely deliver a higher dose of radiation compared to external beam radiation therapy, since the source is located close to the target, so radiation does not need to travel through healthy tissue to have a clinical effect. Additionally, there is excellent dose control with brachytherapy, since there is a steep dose gradient at which the dose falls at a rate of 1/r² with distance from the source. Because you can have a higher dose of radiation delivered, treatment can proceed over fewer fractions of radiation delivery. A main limitation of brachytherapy is that it can only be used in tumors up to 50 to 100 cubic centimeters because of how quickly the dose falls. Additionally, implanting radiation sources can be invasive procedures, and incorrect placement of applicators can create risk of unintended dose distribution. This is a schematic of the dose distribution with brachytherapy in the solid line versus external beam radiation therapy in the dashed line, where blue is the elective target, light red is intermediate risk target, and red is high risk target. As you can see, there is a much higher dose delivered to the high risk target, and this quickly falls off as you go farther away from the target in brachytherapy. There are multiple rates at which brachytherapy can be delivered, such as high dose rate, using iridium-192, cobalt-60, strontium-90, yttrium-90, and ruthenium-106. These are often used in temporary implants with sources that are activated by afterloading machines. There is also low dose rate and very low dose rate brachytherapy, which are often used in permanent implants as a booster to other therapies. These use the isotopes cesium-131, cesium-137, and iodine-125. Another way of classifying delivery is intracavitary, which is in near the tumor, versus interstitial, where the source is in the tumor. Implants can include seeds, wires, and needles. An important development is personalized applicators with 3D printing, which can be customized to an individual's anatomy, allowing the radiation dose distribution to be more optimized and more accurate. Another development has been the integration of imaging into multiple stages of the brachytherapy workflow. For instance, CAT scans are widely used in planning treatment and defining the target region. Between treatment planning just prior to delivery, imaging can also be used as a final check of organ position, as they may have displaced during the half an hour to 2 hours it takes to plan treatment. Excitingly, imaging can be used for real-time guidance during implantation through MRIs, CT scans, endoscopy, and ultrasound. Imaging such as MRIs and CBCTs may also be used for verifying the dose after delivery. Finally, taking images over the course of treatment allows visualization of the tumor shrinking and allows you to adjust the dose and response, which allows brachytherapy to be an adaptive treatment process. In vivo dosimetry is another development that allows you to detect deviations from the treatment plan and detect mistakes such as from equipment failure or dose miscalculation or incorrect applicator placement, and this increases accuracy of brachytherapy. Time integrated point or multi-point dosimetry is most widely used, but it is limited by the influence of dose accumulation of sources at dwell positions. Time-resolved dosimetry, on the other hand, records the dose at dwell positions and tracks sources and measures the dwell time, thus accounting for this influence. Having gone over some of the advances in brachytherapy, we will shift our discussion to its clinical uses, focusing on prostate cancer, breast cancer, and gynecological cancers. Although it is also used in brain, head and neck, anal and rectal, skin, and lung cancers. Brachytherapy is well established for use in prostate cancers as a monotherapy in low-risk cancer or as a boost to EBRT in high-risk cancers. Its main benefit is that it has a lower risk of urinary incontinence, erectile dysfunction, or proctitis compared to surgery or EBRT. Delivery methods include VLDR implants with iodine-125 or LDR implants with palladium-103 or cesium-131 seeds, and these would be a permanent treatment. HDR monotherapy with iridium-192 can also be used as a temporary treatment, although HDR brachytherapy is often used along with EBRT. Another use for brachytherapy is in breast cancer. Standard practice for early breast cancer is breast-conserving surgery, followed by whole breast external beam radiation therapy and an interstitial brachytherapy boost if indicated. An alternative approach is accelerated partial breast irradiation, or APBI, after surgery, where radiation is only delivered to the surgery cavity. This exposes the breast as a whole to a lower level of toxicity. Because radiation must be focused on the tumor bed within the surgery cavity, brachytherapy is used in 30% of APBI treatments. There are multiple delivery methods for APBI, for example, HDR iridium-192, with various delivery methods depending on the geometry of the target region, including interstitial multicatheter, multi-lumen intracavitary balloon catheter, multi-lumen intracavitary cage-like catheter, and single-lumen intracavitary balloon. You can also have permanent implantation of palladium-103 seeds. Two exciting developments are non-invasive image-guided breast brachytherapy after breast immobilization and electronic balloon brachytherapy as well. Finally, brachytherapy can be used clinically in gynecological cancer, particularly cervical cancer, which can use HDR brachytherapy along with external beam radiation therapy. Applicators used include tandems, ovoids, and rings, which are often used along with ultrasound guidance, and larger tumors may require interstitial needles. Brachytherapy can also be used for intermediate to high-risk endometrial cancer, where treatment involves irradiation of the upper third to half of the vagina with delivery through a single channel cylinder or an intravaginal balloon. Finally, going into some of the future directions in brachytherapy, the delivery of radiosensitizing drugs through catheters prior to delivering radiation is promising in that it allows brachytherapy to achieve even higher doses of radiation, thus capitalizing on its strengths. A big problem is that seeds that are implanted may clump together and migrate. However, one approach to fix this is using LDR wires as implants instead. An alternative approach is using biodegradable brachytherapy seeds, which prevent migrating as well. Biodegradable HDR implants have also been developed. A really fascinating idea is using injected radiolabeled self-assembling polymers that don't require surgery, which takes away invasive procedures, which are one of the main limitations of brachytherapy. So in conclusion, brachytherapy is a cancer treatment method in using an internal radiation source. Its main advantage is it can deliver high radiation doses to its target while sparing surrounding tissue. Its clinical uses include prostate cancer, breast cancer, and gynecological cancers, and innovations in personalized applicators, integration of imaging and in vivo dosimetry into workflows, and safe and less invasive delivery methods are increasing the accuracy of brachytherapy and ultimately increasing its clinical value. Again, thank you to the organizers of this conference for the opportunity to be here, and I hope this has been an informative experience.
बोल दे.
discovery.
Uh,
The isotopes are available, and the brachytherapy, which was low dose rate brachytherapy, almost cancer cervix, 6 or 7 days patient has to be into the ward with the isotope. It has been brought down to just 20-25 hours with the medium dose rate, and now with the high-tech HDR, remote control uploading system, the treatment is of minutes, 5 to 8 minutes. Patient can come, take the treatment, go back home within the same day. So that is the HDR brachytherapy. So the evolution has taken place. It is available in India in many centers, including the center in Bikaner, in Jaipur, in Udaipur and all. So again, I hand over the floor to Dr. Madhubala to have her comments.
Thank you, sir. See, radiation biology is very close to my heart. Each time when I listen to my young scholars, I would like to instill this into their minds that you need to understand the mechanisms. You need to do, you may be understanding overviewing the whatever is available in the knowledge domain, which is very important to start with, but don't stop here. They are very young and still they have a long way to go. So if somewhere my message can reach to these young scholars, they should not just be a push button scientist to do this and that without understanding the basics of it. So very important to tie up with the laboratories like INMAS, like any other radiation biology laboratory, carry out these studies jointly to understand more on mechanisms, so that the clinicians have more confidence to deliver these radiation doses for the treatment of patients. Thank you very much.
I fully agree. PhD is not the end of the research. It is the outcome of your research. It's one of the stuff you can say. But the long journey, you have to continue this research till you breathe last. And I always say, teacher is one who is the student all his life, he can be a teacher. If he thinks he is now teacher, nothing to read, he ceases to be a teacher. So you have to be continuously being a student and all. So thank you very much. We go to the another talk from the same group, and it is from Dr. Abhinav, and he will be speaking on overview of immunotherapy. And because immunotherapy again is a very, very wide domain, wide subject. So the group has split it into part 1 and part 2. Abhinav will be speaking on immunotherapy part 1. So the floor is to Dr. Abhinav Shankarati.
Hi everyone, my name is Abhinav.
And my name is Arna.
And we are pre-medical students from Rice University. We are part of the SIR Fellowship Program at Papanini Labs as well. I would like to thank the organizers of the International Conference on Radiation Biology for this opportunity. Our presentation is on cancer immunotherapeutics, and we will dive into the current approaches, delivery strategies, and emerging directions in this vast field. Here is our agenda for the presentation. We will start off with an introduction to cancer immunotherapy. Stimulating the body's immune response to fight against cancer is an exciting advancement in the treatment of cancer. Cancer immunotherapy consists of specific therapies that aim to enable the patient's immune system to find and destroy cancer cells. In 2018, laureates James Allison and Tasuku Honjo won the 2018 Nobel Prize for Physiology or Medicine for their discovery in unleashing our immune cells to attack cancer cells. James Allison discovered that T cell protein, CTLA4, functions as a brake on T cells, and Tasuku Honjo discovered that PD-1, another protein expressed on the surface of T cells, also acts similarly. The seminal discoveries of these two Nobel laureates established a landmark in cancer immunotherapy, improving efficacy in the treatment of many cancer patients. Cancer immunotherapy is now positioned as the first-line treatment for many patients eligible for immune-based cancer treatments. To begin with, the immune system consists of innate immune defenses such as macrophages, dendritic cells, and natural killer cells, which are key players in the immediate tumor immune response. They can engulf and attack foreign cells, or in the case of dendritic cells, present antigens released by tumor cells on its surface. This then activates B cells and T cells. In short, B cells are at the center of the adaptive humoral immune response, as they produce antigen-specific immunoglobulin proteins that are used to target pathogens for destruction, neutralization, or signaling. T cells are important in regulation of the immune system, destruction of pathogenic cells, and sustained immunological memory. When tumor cells die, they release tumor antigens that are uptaken by dendritic cells and presented on MHC1 and MHC2 proteins. CD4 plus helper T cells bind to the MHC2 proteins, while CD8 plus cytotoxic T cells bind to MHC1 proteins. The MHC1 and 2 proteins with presented antigens allows dendritic cells to activate T cells for subsequent immune response. Various chemical signals such as cytokines mediate the entire immune response. The tumor cell has various mechanisms by which it can avoid immune recognition. The first is by not producing an MHC1 protein. This causes the CD8 plus cytotoxic T cell to not be able to bind to the tumor cell and facilitate its destruction. Tumor cells can also produce self-antigens that are taken up and presented on antigen-presenting cells, or APCs, to inactivate T cells. Usually, antigens can bind to tumor cells to neutralize them and signal their destruction. Tumor cells have the ability to engulf the antigen and facilitate their degradation. Tumor cells can also secrete cytokines and chemical factors that inhibit T cells and activate T regulatory immunosuppression cells. The tumor microenvironment can also develop a physical barrier through its extracellular environment that prevents immune cells from binding to tumor cells. As it pertains to radiation oncology, the use of ionizing radiation to kill tumor cells is a hallmark therapy for treating cancer. Ionizing radiation can produce double-stranded breaks in the DNA of tumor cells. This process of DNA damage can lead to double-stranded or dsDNA leakage into the cytosol. This dsDNA accumulation allows it to be used as a ligand for cGAS. cGAS becomes cGAMP, which binds to the STING endoplasmic reticulum protein, which oligomerizes with the kinase TBK1. The STING complex recruits and phosphorylates interferon regulatory factor 3, which translocates to the nucleus and acts as a transcription factor to regulate type 1 interferons and other co-regulated genes. The production of type 1 interferons leads to subsequent dendritic cell maturation and increased CD8 plus T cells in the tumor microenvironment, demonstrating the immune stimulation caused by ionizing radiation. Novel STING agonists are currently being experimented with for immunotherapy. Moving to immune checkpoint inhibitors. Immune checkpoint inhibitors activate or inhibit certain pathways in order to slow down the progression of tumor cell growth. There are 3 essential receptors involved in this: PD-1, PD-L1, and CTLA-4. Cytotoxic T lymphocytes can express PD-1 protein on its surface. PD-1 allows the T cells to detect and bind to tumor cells and facilitate their destruction. PD-L1, the ligand of PD-1, is another surface protein that is expressed by the tumor cell to inactivate T cells by binding to PD-1. PD-L1 is also expressed on antigen-presenting cells. The PD-1 and PD-L1 interaction leads to hindered T cell functionality. Antibodies generated against PD-1 and PD-L1 can reverse the exhaustion of T cells and eliminate tumor cells through lymphocyte activation. CTLA-4 is a protein that is expressed on the surface of the T lymphocyte. The activation of CTLA-4 can produce immunosuppressive response by limiting IL-2 or interleukin-2 production and NFKB signaling, as well as immunosuppressive activation of T regulatory cells. Thus, CTLA-4 is a negative modulator of immune function against tumor cells. Unfortunately, checkpoint inhibition does have some immune-related adverse effects. The promise of ICI treatment is great despite these challenges, as combination therapies and novel approaches to cancer treatment continue to push the envelope for ICI treatment. Targeted antibodies, specifically monoclonal antibodies, are a type of antibody that are made in the lab and have the ability to target specific cancer antigen. They also have a variety of roles, such as neutralization of pathogens, opsonization for phagocytosis, the recruitment of T cells, and various others as shown in this diagram. Affinity maturation is a process that increases specificity and antibody activity against the target antigen. This process occurs in all monoclonal antibody productions. Monoclonal antibodies are produced through CDR grafting, antibody phage displays, and even human antibody-producing mice. One type of monoclonal antibody is antibody drug conjugates, which includes a cytotoxic payload connected to a chemical linker. They deliver a toxic payload to cancer cells to improve chemotherapy. And this can be seen as well with radioactive particles, immunotoxins, and siRNA. Some negative side effects include cell toxicity and a decrease in efficacy of the therapy. Bispecific antibodies are another type of monoclonal antibodies that make T cell effector cells and NK cells attack tumors. They specifically bind to both T cells and tumors. Many of these antibodies work as signal blockades through the anti-PD-1 and anti-CTLA-4 treatments. Next up, we have cytokines, which are small proteins or glycoproteins that send signals to other cells in response to differentiation and cellular stress. They bind to target cells and trigger intracellular signaling pathways, leading to modified gene transcription. Some of their limitations include a short half-life, autoimmune attacks, and vascular leakage. Overall, cytokine therapy has a low rate of effectiveness, but the interleukin-2 and interferon alpha cytokines have shown mild benefits for anti-tumor efficacy, leading to FDA approval. Interleukins generally work to enhance cytotoxic T cell function, as well as other anti-tumor lymphocytes. Similarly, interferon alpha activates immune cells to produce an anti-tumor effect. The functions of cytokines are diverse and wide-ranging. Cytokines can also be engineered to improve delivery and efficacy. Molecular engineering includes immunocytokines, which consists of the fusion of cytokines to full-size antibodies or antibody fragments. Site-directed and combinatorial mutagenesis is used to directly engineer cytokine binding sites with altered receptor specificity. And we also have de novo protein design to create mimics of cytokines with higher stability and specific affinity. Chemical engineering of cytokines can be done through polyethylene glycol, synthetic polypeptide, and extracellular matrix-based conjugation. Both molecular and chemical engineering can increase receptor specificity, half-life, and stability. Thus, limiting adverse effects and improving localized immune activity is a result of engineering cytokines. Cancer vaccines are adjuvants that enhance the immune system's response to the presence of an antigen. They're prophylactic vaccines that are administered to healthy individuals and therapeutic vaccines that are administered to individuals who are combating a current disease in order to slow its progression or stop it altogether. Tumor-associated antigen, TAA, are self-antigens that are aberrantly expressed in tumor cells, while tumor-specific antigens, TSA, contain antigens that are encoded by cancer mutations. Neoantigens are TSAs that arise from somatic DNA alterations in cancer cells, and they are unique to patients, and thus require a personalized approach by genome sequencing of the patient's cancer cells. Next, the neoantigens are predicted, and a vaccine is produced to express predicted neoantigens that can promote lymphocyte activation against tumor cells. Oncogenic viral antigens are derived from oncogenic virus surface proteins for potential tumor rejection. There are various mechanisms of cancer cell delivery and development. DNA and mRNA vaccines are taken up by various antigen-presenting cells and are translated to induce antigen expression. Due to nuclear delivery barriers and low immunogenicity, DNA vaccines are often unsuccessful. mRNA-based vaccines can directly induce antigen-presenting cells to express antigens. They also have the advantage of not having to enter the nucleus and only having to enter the cytoplasm. Whole cell vaccines include modified antigen-presenting cells. For example, these vaccines can be made from dendritic cells collected from patients that are engineered to express tumor-associated antigens, thus activating T cells directly to attack tumor cells. The advantage of using peptides in comparison with whole cell lysates is that it delivers only the epitopes of interest to the immune system, rather than overloading immunological pathways with irrelevant antigens that may compete with relevant epitopes. Overall, cancer vaccines have relatively low efficacy, but they still hold value in prophylactic and therapeutic cases.
Another type of immunotherapy is adoptive T cell therapy. Adoptive T cell therapy harnesses the patient's own immune cells to produce a tumor-specific response. This can include tumor-infiltrating lymphocyte therapy, engineered T cell receptor therapy,
or chimeric antigen receptor T-cell therapy. Tumor infiltrating lymphocyte therapy harnesses immune cells which are present in the tumor microenvironment. These TILs can be effectively isolated from resected tumors using techniques such as single-cell RNA-seq and mass cytometry. Once isolated, these cells are expanded ex vivo and are then infused into the patient along with a lymphodepleting regimen and large doses of T-cell growth factor, IL-2. This lymphodepleting regimen is necessary to promote TIL proliferation by reducing nearby Treg cells and decreasing competition for cytokines. TIL therapy that does not modify cells genetically can still be highly effective in cancer, such as metastatic melanoma. There are multiple ways to genetically modify tumor-infiltrating lymphocytes. Retroviral transduction, zinc finger nuclease, TALEN, and CRISPR-mediated knockout methods can all be used to provide targeted responses with limited off-tumor side effects. Additionally, specificity assays can allow for the expansion of tumor-specific TILs to increase a directed response with decreased toxicity. There is a lack of effective treatment results with TIL therapy in non-melanoma cancers. However, new approaches, especially combinatorial approaches, have shown promise in various studies to improve TIL efficacy for other types of tumors. T-cell receptor or TCR engineering approaches use the native or minimally modified TCRs to recognize specific tumor epitopes. Ideally, antigens used in adoptive cell therapy must be tumor-specific in order to avoid the off-tumor toxic effects that are seen when using tissue differentiation antigens, since these antigens are also present in normal cells. Out of the many mutations cancer cells induce, very few of these mutated antigens can effectively bind to peptide MHC complexes and be recognized by T cells. Neoepitopes, which are expressed sufficiently by a cell's MHC complex and which prompt T cell responses, are considered neoantigens. Targeting neoantigens can allow T cells to more precisely recognize non-self antigens with reduced risk of autoimmune complications, and this approach has been shown to produce near total regression in various cancers. Proteomic methods have shown that there exists a wide variety of tumor-specific antigens which were previously unidentified with only whole exome sequencing or RNA-seq methods. Furthermore, current methodology involving deep learning has enabled researchers to use a de novo peptide sequencing workflow to improve the identification of neoantigens from mass spectrometry data. These sequencing procedures can identify sequences that form new epitopes that may be recognized by T cells. Then TCR-specific neoantigens can be identified. Gene editing methods such as CRISPR-Cas9 can be used to knock out endogenous receptors, resulting in higher expression and function of transgenic TCRs, stronger anti-tumor response, a 1,000-fold increase in antigen sensitivity, and efficient redirection of CD8+ and CD4+ T cells to malignant cells. CAR T cells are developed by genetically engineering patient-derived T cells to exhibit chimeric antigen receptors, or CARs. These CARs are specific for antigen present on the surface of tumor cells. They have an extracellular protein portion that is derived from an antibody and an intracellular protein portion derived from T cell signaling proteins. These receptor molecules recognize antigens independently of MHC. This is advantageous because cancer cells usually evade the immune response by decreasing MHC-associated antigen presentation. Upon administration into the patient's bloodstream, the CAR T cells detect the tumor antigen and promote apoptosis. CAR T cells can also target inhospitable tumor microenvironments and revive exhausted T cells. The efficacy of CAR T cell therapy is based on targeting a specific antigen on the cancer cell, such as CD19 antigen for B-cell malignancies like acute lymphoblastic leukemia. If the target antigen is not present, the treatment will not work. CAR T cell therapy may have toxic side effects that affect the body in different ways, most commonly through cytokine release syndrome and neurotoxicity. This toxicity is more severe than that of TCR therapy. Another limitation to the gene-edited T cells is exhaustion of these cells when in the tumor microenvironment. The exhaustion of the T cells leads to cancer immune evasion. T cell exhaustion also involves changes in the cellular metabolism and aberrant transcriptomic and epigenetic profiles. However, CRISPR seems to have a great potential for reversing exhaustion of T cells in the tumor microenvironment. While exhausted T cells can be restored, the epigenetic landscape will not be altered significantly, causing the restored T cells to prevent the formation of memory cells. Current studies show the therapeutic promise of epigenetic programming for exhausted T cells. Overall, the FDA has approved 5 CAR T-cell therapies in the past 5 years to treat leukemia, lymphoma, and the role of CAR T-cells for cancer therapy is expected to advance over time. Natural killer cells, or NK cells, are innate immune cells with potent cytolytic activity against tumors. NK cells are a promising platform for cancer immunotherapy because their availability and acceptable safety profile in the allogeneic setting. Uniquely, NK cells show the ability to recognize and kill tumor cells regardless of HLA-1 expression, which is a mechanism of immune evasion, without recognition of tumor antigens and also without recognition of MHC molecules. Thus, NK cells can be employed to treat multiple cancer patients and tumor types. The NK cancer cycle consists of 4 main steps. First, NK cells are recruited into the tumor microenvironment. Next, NK cells recognize the cancer cells and undergo full activation. Then, NK cells kill the cancer cells locally and systemically. And finally, NK cells orchestrate innate and adaptive immunity. And the cycle repeats. Various signaling molecules can positively and negatively regulate points within the NK cell cancer cycle. Homogeneous NK cell lines can be established using the NK cells of patients and donors, as well as by the differentiation of induced pluripotent stem cells. Recent research studies develop promising strategies to improve the expansion quantity and anti-tumor functionality of NK cells. These approaches use cytokines and antibodies to improve the efficacy of NK cells and modify NK cells with a genetic engineering to generate chimeric antigen receptor, or CAR NK cells. Furthermore, the future genetic manipulation approaches using CRISPR-Cas9 or TALEN present opportunities to improve the activity of NK cell-based therapeutics. Limitations of immunotherapy include adverse immune side effects and non-specific inflammation that can be detrimental to patients' health. Furthermore, only a small set of patients respond to immunotherapy interventions. Delivery platforms have been shown to improve safety and decrease toxicity by allowing for the controlled and localized delivery of immunotherapeutics to the site of the tumor. One major delivery technology approach is using nanomedicine. For example, nanoparticles can mediate the delivery of vaccines, and nanodisk-based vaccines can greatly improve the efficacy of co-delivery, the efficiency of co-delivery of antigens and adjuvants to lymphoid tissues, thus inducing dendritic cell maturation. For example, nanoparticles can mediate the delivery of vaccines, and nanodisk-based vaccine can greatly improve the efficiency of co-delivery of antigens and adjuvants to lymphoid tissues, thus inducing dendritic cell maturation. Nanoparticle-mediated delivery targets multiple inhibitory signals in the tumor microenvironments. Implantable scaffolds are biomaterials that can be preloaded with a variety of chemical reagents, biological factors, or cells. These scaffolds are typically implanted in subcutaneous or resected sites through surgical procedure. The bioactive agents can be controlled to release in the implanted scaffolds, and thus immune cells are generally recruited to access these scaffolds. Another delivery technology are injectable biomaterials, which include hydrogels and cryogels. The advantages of these injectable biomaterials are that they require minimally invasive procedure, and the materials can be positioned anywhere the needle can reach, thereby avoiding tissue damage and other inflammatory wound complications. The matrix binding molecular conjugates are one other class of delivery methods which have been developed to accumulate within and around tumors. Basically, the conjugates are more localized in extracellular matrix around the tissue, leading to delayed tumor growth and extended survival. While novel delivery strategies have potential for cancer immunotherapy, there are still limitations that need to be considered. The size of the nanoparticles influences their pharmacokinetics in vivo. Nanoparticles less than 200 nanometers in size can move freely in the lymphatic circulation to deliver antigens and or adjuvants, and therefore increase the likelihood of activating antigen-presenting cells. The translation of nanoparticle-based delivery requires an accurate assessment of their toxicity. It is not clear whether nanoparticles increase autoimmune reactions in addition to their stimulation of the immune system. Furthermore, nanotechnology could prove detrimental to the clinical translation of nanoparticle-based immunotherapy due to increase in cost and complexity to manufacture and commercialize these agents. Additionally, degradation of biomaterials such as scaffolds and hydrogels may still trigger acute inflammatory reactions. On the other hand, implantable scaffolds are prone to breakage and require surgery to implant in subcutaneous areas. Moreover, these implants are limited to the accessible location of the surgical procedure and cannot be easily implanted anywhere it is desired. Combination therapies that combine agents with distinct mechanisms of action have enhanced the success of various cancer treatments. For example, ipilimumab and nivolumab combination therapy presented a significant survival benefit in patients with metastatic melanoma and advanced renal cell carcinoma. Another attractive synergistic combination therapy is combining radiotherapy with blockers of the PD-1 axis. Specifically, dual checkpoint blockade and radiation checkpoint polytherapy represent promising avenues for synergistic therapeutic responses, as these combinations of drugs display unique and complementary pharmacodynamics. Furthermore, emerging research is directed at newly discovered negative regulators of T-cell activation as adjuvant cancer drugs. Overall, cancer immunotherapy has emerged as a powerful set of methods for treating cancer by harnessing the patient's own immune system. Many delivery technologies can be used to accommodate a variety of immunotherapeutic agents that are selected based on patient-specific targets, leading to personalized treatment. These cancer immunotherapy success stories indicate how a bench-to-bedside approach built on a solid science foundation can be successful in combating cancer. Thank you so much for listening to our presentation. We would like to once again thank our mentors, Dr. Papaneni and Dr. Verma. We are also grateful to the organizers of the International Conference on Radiation Biology for allowing us to speak today. Thank you.
Thank you, Abhinav, for your overview on immunotherapy. As you have seen how immunotherapy can be useful, the research being put on also towards cancer vaccination, and also how nanotechnology can combine with this thing for conquering the cancer. Till date, we were just saying that radiotherapy, surgical oncology, and medical oncology are the only treatment, but immunotherapy in many cases like the prostate, breast, and other melanomas is being used, but the research is towards combination of the therapies and how immunotherapy can work. And thanks to Dr. Rao Patineni, and it is 3 a.m. at Brantford in USA, and he is live, he is interacting. That shows the interest, that shows the nurturing. He has nurtured, he has mentored these scientists, young scientists, budding scientists in the research. So thanks to his lab and the Dr. Papi Nemi and these activities. Do you want to add few things, madam?
Nothing beyond, sir. I would like to congratulate Dr. Rao Pepineni. He's been associated with society for quite some time, and in each meeting, he has been giving wonderful contributions. We would love to have him with us for all the times to come, and maybe he can pick up some of these students from these laboratories as well, you know, if he is there in person. So thank you very much, Dr. Pat. And you may proceed, sir.
Wait. You wanted to say something?
I said you may please proceed on. Thank you.
Thank you. Thank you. So now the last talk of this group is from Arnav, and he will be explained. Okay, join. So now we have done with this all 5 invited talks, starting from Dr. Seema Gupta to 4 from the Patinini lab from all USA. And wonderfully, they have covered the research going into the field. And this is the youngsters who can take forward the initiative, what is happening, and how this can be taken forward. With this word, thanking also Dr. Madhubala from at a distance joining. And I know, being a director, I was looking at signing the files also going on, work is also going on, here also listening very attentively. So everything is going on. That is how the devotion for the science, devotion for the profession, for the society. Thank you, madam, for joining with us. Thanks to the organizers for giving me the chance to chair this session. And along with me, my colleague Dr. Smita Jain is there, who will be reporting about summing up the session, what has happened. So I hand over the mic to her.
I would like to say something. You see, COVID cases are on rise here. And we have recently established a BSL-3 facility, first of its kind in Uttarakhand, which is a container-based facility and can be towed away. So now almost every day I am getting requests that they want more testing to be done. And what you were seeing as files, they were actually the approvals for going ahead for the testing. So the patients are applying. It's a very sad situation right now. About 30% of my staff is infected. I'm so sorry, but this is what is the reality today. And this is what prevented me from reaching out. Bikaner is very close to me. Dr. Purohit knows, and the entire team knows that I would always love to be there, interact with young scholars and the faculty, and of course principal and the vice chancellor. And sir, I missed the opportunity to being with Dr. Chogle also, but this is the limitation of the time. And let us see. See, radiation biologists have already infiltrated into the COVID therapy also. So, so thank you so much. This is all I had to say. Namaskar. Very good session. Very informative.
Honorable Chairpersons, Dr. Madhubala Ma'am, President ISRB and Director DRDO, Dr. Arun Chogle, Professor and Head, Department of Radiotherapy, SNS Jaipur, Convener ICRB, Dr. R.K. Purohit, Organizing Secretary, Dr. Aruna Chakrabarti, Co-Convener, Nikti Srivastava, all the delegates, participants, and colleagues, very good afternoon to all of you. It is my real pleasure to present to you the report of International Conference on Radiation Biology, Day 1, Session 1, on implications of management of cancer. I would like to thank Dr. Seema Gupta, Dr. Krishna Koka, Dr. Sahiti, Dr. Abhinav, and Dr. Arnav for joining all the way from USA. All the papers were very well presented and very informative with the description of latest technologies. I will summarize my report in total. Radiation therapy can be broadly classified into brachytherapy and external beam therapy. Brachytherapy or internal radiation therapy treats prostate, breast, head, neck, and other cancers. Doctors place tiny radioactive pellets inside or next to the tumor. The pellets give off radiations that destroys cancer cells. The treatment spares surrounding tissues and organs. External beam radiation therapy comes from a machine that aims radiation at your cancer. It is a local treatment, which means it treats a specific part of your body. For example, if you have cancer in your lung, you'll have radiations only to your chest, not to your whole body. Numerous preclinical studies have suggested that combining immunotherapy with radiotherapy could be a promising strategy for synergistic enhancement of treatment efficacy. With the development of the field of cancer immunotherapy, the focus of treatment has shifted from treating the disease site to treating the specific tumor's biological characteristics and its interaction with the intrinsic immunological ability of the patient to combat the disease. A patient's immune cells can be stimulated and activated in vivo through the application of immune checkpoint inhibitors, targeted antibodies, cytokine therapies, and cancer vaccines. As earlier said by Dr. Arun Chogle, it is not only radiations and immunotherapy. In addition to it, our lifestyle, eating habits, and most important, positive attitude and meditation also help to fight against cancer and other diseases. And I think this conference including latest research papers, will surely surely benefit the society, and it means it will go beyond words and not just discussions. Lastly, I would like to congratulate Dr. Purohit and his entire team for organizing this international conference in these adverse conditions, and my best wishes for its successful completion. Thank you all.
Thank you, Dr. Thank you, Dr. Madhubala, ma'am. Is working.
Thank you, Dr. Madhubala, ma'am. Dr. Arun Chogle, for chairing the session. I know Dr. Madhubala is not feeling well. Even then, she has spared. She has been with us this morning. Many thanks, ma'am. Dr. Chogle, sir. Many thanks. We have a very hectic program for you. In the morning, you were the chief guest. Now you are in the session. Now, ladies and gentlemen, dear students, after lunch, there will be first talk. Will be delivered by Dr. Arun Chogle. Please be with us. He remains available here. I would request Professor Abhi Salman come on the dais, and Professor Madam Veena come on the dais. Dr. Sundajan also to present memento to Chairperson Dr. Arun Chogle. Sometimes Veena forgot. Dr. Veena Purohit, she is in MS College. She is a professor of botany. She is a please. Manisha, you also come. Manisha, ji. Just yeah. Thank you. So we conclude this session. You all are invited for lunch. Have lunch, quick lunch, and come back to the hall. Please be here with us, because we have very nice culture program today evening. If we have to finish the sessions as it is possible.